Hello, everyone. Welcome, and thank you for joining us for the sixth lecture of the third edition of the lecture series Current Topics in Heritage Science, organized by emerging professionals in the frame of E-RIHS Academy. My name is Fabiana Di Gianvincenzo. I am a postdoctoral fellow at the Rijksmuseum in Amsterdam, and I will be moderating today's lecture titled Pyrolysis-gas in Heritage Science, an important investigative tool for the analysis of organic materials. You can ask questions via the Q&A functions at any time, and they will be answered by the speaker at the end of the lecture. Please note that the lecture is recorded and it will be available on the E-RIHS YouTube channel. If you experience any technical issues, let us know through the chat. After the lecture, you will also receive a link to a survey. Please take a few minutes to share your opinion with us. Our lecture today will be given by Jennifer Poulin, Senior Conservation Scientist at the Canadian Conservation Institute in Ottawa. Her educational background is chemistry, and her main research interests center on the development of direct inlet pyrolysis-gas chromatography-mass spectrometry for use on heritage microsamples, and the use of GCMS to study a range of organic materials, from natural and synthetic dyes to Canadian amber, paint-binding media, archaeological objects, pesticides, modern materials, and varnishes and lacquers. Jennifer is a board member of the Mass Spectrometry and Chromatography Users Group, or MASC. She is currently planning the Dyes in History and Archaeology 44 conference, the DHA, that will be held in Ottawa in October 2025. And now, without further ado, Jennifer, thank you very much for joining us today, and the floor is all yours.
Okay, I'm going to share my screen and get started. And how does that look?
Perfect.
Okay. Well, thank you very much to the HS Academy for inviting me to speak today about pyrolysis, gas chromatography, mass spectrometry, and heritage science. Before I begin, there are many scientists in our field who are truly experts in PyGCMS, and I still turn to some of them when I have questions. And I want to stress the importance of not isolating yourself. Stay connected, reach out. In my experience, we're so lucky to work in a field that is supportive and generous. So, PyGCMS. I'm passionate about the sheer amount of information that this elegant technique can provide when thought and care is given to the way it's used. So let's get started. I'm speaking with you today from Ottawa, Canada, where I work as a senior conservation scientist at the CCI. The land on which CCI is located and upon which this work has been carried out is in the traditional and unceded territory of the Algonquin Anishinaabe people. CCI is an operating agency of the Department of Canadian Heritage. We currently have about 60 to 70 permanent and temporary staff in roles from science, treatment of objects, to risk assessment, heritage places conservation, scientific imaging, along with our incredible support staff for the library, communications, building management, and health and safety. So it's a very good place to work. Here I am in grad school in 1994, 31 years ago, just as I was emerging into my career. This is my first GC. It's a Shimadzu A8 with a column that I packed myself and peaks that I measured with a ruler. My first job was the analysis of food and organics for the Canadian Border Services Agency, and I left that job in 2003 to begin my work at CCI in the Conservation Science Division. Typically, my work involves analysis of paint and varnish, natural resins, archaeological residues, and dyes. And at the CCI, I focus on just one type of instrument, GCMS. There are many ways that we can use that instrument, and today I'm going to focus on sample introduction by pyrolysis. I thought I'd begin by showing you a typical protocol for analyzing an unknown sample where the PyGCMS would fit into the workflow. At the CCI, we work in teams with one person leading the analysis request and the rest of the team joining in when required. I'll begin with the left-hand path, where the sample is homogeneous and not layered. So a typical first step for an unknown sample of this type would be FTIR spectroscopy. IR spectroscopy gives us a good overview of the entire sample. It's non-destructive, and the sample can be retrieved for further analysis. If the sample is found to be inorganic or a mixture of organic and inorganic components, the sample can be removed from the diamond cell and adhered to a planchet using carbon tape for further analysis of the inorganic components using SEM-EDX, Raman, or X-ray diffraction. When the material is primarily organic and further analysis is required, the sample can be retrieved. For instance, if we need Raman to look for organic pigments that weren't fully identified with the IR. If not all the components have been identified in the first fragment of the sample with those methods, a second piece of the sample can then be examined using additional techniques. For instance, if the IR spectrum of a varnish indicated that it was an acrylic copolymer, further analysis using PyGCMS would provide details about the polymer, as well as low concentrations of additives. For multi-layer samples, a typical protocol would start with separating the sample into two parts. One fragment is mounted and as a cross-section to study the stratigraphy and analyze the layers using techniques that can be undertaken directly on the cross-section. And you can see them listed here. Sometimes these methods don't allow all the components to be identified. And if this is the case, a second unmounted fragment is used to separate the layers of interest, like PyGCMS, for example, and that can be because PyGCMS analysis can't be carried out directly on the cross-section, we have to excavate the layers. So now that you see where PyGCMS is usually performed at CCI during multi-analytical investigations, which is usually at the very end, let's dive into its history, technology, and use. So PyGCMS was first used to analyze paints and coatings in 1965. In this case, they used a resistant heated platinum coil for the pyrolyser, and it was coupled to a flame ionization detector. Then in 1966, Simon used pyrolysis coupled to GCMS. The pyrolyser type that he employed was a Curie point. And soon afterwards, Nathan Stolo, who was the founder of CCI, along with George Rogers, first published the use of PyGCMS in the analysis of works of art. Fast forward nearly 2 decades, and many more publications later, to this article in the Journal of Analytical and Applied Pyrolysis by Shvedrinsky, Wampler, Indictor, and Baer. Here they discuss analysis of most classes of materials that we find in cultural heritage, art, and archaeology. And finally, we come to what I think of as the modern era of PyGCMS, with one of the main authorities in the analysis of modern paints, written by Tom Lerner. In this thesis, he discusses the thermal degradation reactions of acrylics, vinyl acetate, polyurethane, silicone, additives, and synthetic pigments. His book is one that I still turn to, and he was kind of leading the charge in 2004 for modern paints. My most beloved book is by Tsuge Otani and Watanabe. This text is a massive index of pyrograms, and thermograms, and marker mass spectra for 165 standard synthetic polymers. I love this book so much. I recently found a sentence by the authors in the preface. It says, the authors would be in supreme bliss if this data book could contribute to move up the status of PyGCMS in the field of polymer characterization. That was written in 2011. In the past 2 decades, there's been many publications that feature PyGCMS using a variety of pyrolysers and giving us important hints and tips and marker mass spectra for analyzing cultural heritage objects. What an era to be working in. Let's have a look at pyrolysis as a sample introduction method. So the sample is heated at a high temperature, over 350 degrees Celsius, and that causes thermal decomposition. The atmosphere is inert. There's no oxygenation reactions that can happen. And we see scission reactions instead that can be categorized as random, side group, or monomer reversion. I'd like to point you to this book by Wampler if you're interested in learning more about the different thermal decomposition reactions for different types of polymers. It's a really good resource. How do you choose a pyrolysis temperature? Optimum pyrolysis temperatures are usually between 350 and 750 degrees Celsius, although they can go higher. You can determine the optimum pyrolysis temperature for a sample by examining the thermal analysis data, which shows the temperature at which the bulk of the substance is going to undergo thermal decomposition. And thermal decomposition can occur in multiple steps. In this case, I've drawn one for you. The green graph line is, excuse me, the thermal analysis graph. And if we take the first derivative of that graph, you get one that's a little bit easier to read. This is the derivative curve of the TGA. And on this curve, we can see that we start to get some decomposition occurring at about 260 degrees Celsius. The bulk of this imaginary substance is decomposing at 360 degrees Celsius, and it's pretty much completed its decomposition at 450 degrees Celsius. So you can do this in your own lab using known reference materials. You can also do this sort of thing using evolved gas analysis. There's many publications that contain thermal analysis that you can also use to help determine the optimum temperatures when you have an unknown substance. In gas chromatographic separations, two of the most important factors are the mobile phase and the stationary phase. The carrier gas flows along inside the column, bringing the volatilized pyrolysates with it. As the sample mixture flows through the long column, which can be 15, 30, or even 60 meters in length, components begin to separate out based on their molecular masses and their interactions with the stationary phase coating inside the column. I generally use an all-purpose 5 ms stationary phase, which I find is good for most of my compounds. But if you're using something else and it's working for you, keep going. There's many ways in which we can make PyGCMS work for us, and we all have our preferences and different types of samples. When the separated sample reaches the mass spectrometer, it moves from a zone of high pressure to a zone of high vacuum. As each component exits the column, the molecules are immediately bombarded with an ionizing electron stream, and this bashes the molecules apart into smaller pieces, and each piece has a positive charge. In EI mass spectrometry, the electron stream is a constant 70 electron volts. Every time a molecule is subjected to this, it is going to break apart in the same manner, just like a fingerprint. These charged fragments get filtered in the mass analyzer, then they're amplified and detected in the ion detector according to their mass to charge ratio. And what we then see is a chromatogram and a fingerprint mass spectra for each peak. So here's a little chromatogram. The green peak is made of a compound that fragments like this in the mass spectrometer. This compound is 3-pentanone, and it has a molecular mass of 86. When it's hit with an electron beam, it produces 3 main ion fragments. So 2 are formed from breaking the bonds between the carbonyl and the ethyl chain. And then we also see the molecular ion that occurs from the loss of a single electron from the entire molecule. 3-pentanone will always fragment like this during EI ionization, and this mass spectrum is its fingerprint. Pyrolysis heating rates can be fast or slow. Flash pyrolysis is almost instantaneous. So we're talking about 10 degrees Celsius per millisecond, whereas a slow pyrolysis has a heating rate that may take up to a minute or two before reaching the final temperature. Some pyrolysers are capable of performing both flash and slow pyrolysis, and I'm going to show you 4 different types. We're going to look at some rough schematics now. I drew these myself to avoid any copyright infringements, so please take them with a grain of salt and don't hate on them. On each side, there's a QR code that'll take you to an instrument manufacturer's website, so you can learn more about that type if you want to. Please do follow the codes. You will undoubtedly learn more than I can tell you in this really short talk. So this is the induction type pyrolyzer called a Curie point. It heats it heats a foil using a high frequency magnetic field, and depending on the Curie point of that foil, that's the temperature that you get. In our field, Curie point analysis is generally performed at 610 degrees Celsius. The advantages are you get instantaneous heating and it's very precise. Those foils are calibrated. The disadvantages is that it's not flexible. Depending on the Curie point temperature of the metal of the pyrofoil, that's the temperature that you achieve. However, there are different pyrofoils that are available, so you can get a few other different temperatures. It does require split mode operation, so I think you're going to be looking at samples that are minimum of 25 micrograms. The filament type pyrolyzer uses resistance to heat a sample tube with a platinum filament. So there's a lot of advantages to this. You have a wide range of pyrolysis temperatures, and you're also capable of doing multi-step heating. It can operate in flash mode, but you can also slow it down and operate in slow mode. It is pretty easy to install it and remove it from the inlet. So once you convert it to PyGCMS, you can take it off again and use the instrument in GCMS mode. And these are compatible with many different types of GCMS brands. It does require split mode operation. So we're looking at 25 micrograms, probably as the smallest sample. And I found in my use that the coils were pretty delicate. It was a little bit tricky sometimes to use. But some of the newer types have this drop-in sample chamber, which looks really great. Here's another type of filament pyrolyzer where the sample in a liner is inserted above the inlet in a liner-in-liner configuration. So the sample is pyrolyzed and then it's cryo-focused immediately before entering the GC. So the advantages are that it has very precise heating like before, and it can be operated slow or fast. This liner-in-liner pyrolysis and analyte transfer means that there's no transfer lines or valves, and it's very sensitive. In fact, it can be used in splitless operation. So here we're seeing that samples can be analyzed that are less than 10 micrograms. It is usually compatible with most GC brands, and there's also an auto sampler that you can add on. I think the disadvantage is that because of the cryo-focusing that is required with this one, you do have a little extra setup. When you're installing it, it might be more difficult to remove to convert back to GCMS. And finally, this is the furnace type pyrolyzer. It heats a sample by dropping a sample cup into a preheated furnace. So there's many advantages. And this type of pyrolyzer is very well used and popular. It's capable of multi-shot analysis, so you can have multiple heating steps. In fact, I believe with the Frontier pyrolyzer furnace, the sample cup can actually be removed from the heating zone after it's pyrolyzed or been heated for that one time, and then you can put it back in for another heating zone, which I think is really great. It's auto sampler compatible, and it's very compatible with many different types of GC. It is a little bit more difficult to install and remove the pyrolyzer furnace. In the ones that I've seen set up, the system becomes dedicated to PyGCMS. It does require split mode operation and slightly larger samples. On the Frontier website, it suggests 100 micrograms would be the minimum, but I think you could probably push this to 25. Well, pyrolyzers that can only perform slow pyrolysis rely on the heating rates of the GC inlet, and these can be referred to as direct inlet types. The sample is inserted into the GC a few millimeters above the head of the column, and then the inlet is heated rapidly, but not instantaneously, to a pyrolysis temperature. This is the pyrolysis method that I now use at the CCI, and I know that it's also used at the Getty, the Art Institute of Chicago, and the National Gallery in London. For me, the most important advantage is the size of the sample. Because pyrolysis occurs inside the inlet, it can be operated in splitless mode, using just a few micrograms of sample. I've even scraped the sample from the FTIR microscope diamond cell and analyzed it using this method. So most of our samples that we get at CCI are very small. Other advantages are that it's very compact, and it uses because it's inside the inlet, it uses the instrument's own pressure controls, and it's virtually leak-free. I haven't had a leak in 13 years. I've been using it. It's got lots of disadvantages. It's not capable of flash pyrolysis, so the The Optic 4 by Shimatsu can heat to 400 degrees Celsius maximum at a 60 degrees per second heating rate. The Agilent thermal separation probe, the one that I use, has a 450 degrees maximum temperature, and it heats at a rate about 15 degrees Celsius per second. Currently, this method isn't as published as other pyrolyzer types, and it's only, if I understand correctly, it's only compatible with the instrument brand that makes it. We've talked a lot about samples size already, so how small is small? Over the decades, as technologies have improved, we've been able to vastly decrease the amount of sample that we require. In this photograph, you may be able to see these little specks. A sample this size weighs about 20 micrograms, and that's pretty typical for CCI and enough for all of the instrumental analysis that we're going to carry out. Most of our samples are sent to us from across Canada, and we don't have the opportunity to resample. For PyGCMS, I'll use just a few micrograms, and it looks like this in the sample tube. This is a zinc white oil paint. Here's the chromatogram that I actually obtained from that spec, derivatized with TMAH and pyrolyzed at 450 degrees Celsius. It was run in splitless mode, and it shows some excellent signal to noise. I wanted to show that very small sample size is now an option for PyGCMS. Let's get into some sample analysis. For me, it all began with amber. Kate Helwig and I had just finished a project on a new type of amber from the high Arctic in Canada, and we published it after flash pyrolysis using a filament type pyrolyzer. It was a CDS PyroProbe 5000. We bought a new GCMS. Then the application scientist at Agilent had an interest in heritage science, and he asked if he could include a multimode inlet and a thermal separation probe with the installation. And since it was free, I said yes. I didn't really give it any thought until it was being installed, and I learned that the multimode inlet could heat to 450 degrees Celsius, which sounded like pyrolysis to me, because at that point I was obsessed with amber. The optimum pyrolysis for amber is 480 degrees Celsius, and I thought 450 is pretty close to 480. The moment those installation guys left my lab, I immediately analyzed some Canadian amber, and it worked really great. I could even use smaller samples, which was really important. So due to the larger sample requirements that I had for my pyroprobe, there were many heritage object samples that I just couldn't analyze because we didn't have enough for my technique. All right, let's look at this amber. Here's what the class 1D amber looked like when it was flash pyrolyzed with the pyroprobe at 480 degrees Celsius. There is a significant peak of succinic acid, and here we can see that there's pairs of labdane pyrolysates. There's 4 of each. There's also monoterpenes, abiotanes, and pemaranes present in the amber, and flash pyrolysis here with the pyroprobe gave a really great picture of the building blocks for this amber. Kate and I published it, and it was great. I was really happy, and then I got the TSP. And I decided to run all the amber references again at 450 degrees Celsius using slow pyrolysis. And here's the same class 1D amber sample run using slow pyrolysis with the TSP at 450. And we see some similar results. You see the significant peak of succinic acid, pairs of labdane pyrolysates, monoterpenes, pemaranes, and abiotanes. However, there's something really exciting and different that's happening at the higher retention times. And before I show you, I want you to remember these 4 labdane alcohol pyrolysates right here, because they're important. These 10 peaks at the end of the run represent the 10 possible combinations of the 4 alcohol pyrolysates esterified with succinic acid. Using the slow pyrolysis, larger fragments of the polymer were remaining intact. These 10 peaks represent cross-linking of the amber polymer with succinic acid, and we can see clearly for the first time how the polymer was put together. Similar clusters of peaks occur with Baltic amber, which also contains succinic acid. So succinic acid cross-linking in Baltic amber had been hypothesized from the FTIR absorbance bands in 1972, but it wasn't proven until these experiments at the CCI with slow pyrolysis. Well, now let's look at a sample of a 19th century paint that has a few surprises hidden inside. This object is a musical manuscript that's owned and used by John Buttery in the 19th century. The little painted pictures decorate song sheets used for a military fife and drum band. The sampling of this manuscript was delicate. It's thin paint layers on paper. So I used a technique called magnesium oxide microswabbing, or MOMS, and it was developed by Art Proanjo Gabor at the Rijksmuseum, specifically for sampling thin paints and inks on paper. So the tiny particles are removed from the surface using the shaped tip of a thin magnesium oxide rod. And that I place directly into a pyrolysis tube, treat it with TMAH, and the whole thing is pyrolyzed. The MOM sticks are inert and they can be reused indefinitely. So for this study, the MOM sampling and then the subsequent PyGCMS was useful for the characterization of the binding medium, as well as the identification of certain organic pigments. In the yellow paint, I identified an oil binder, which is probably from a marine source like fish. And the presence of alkylphenyl alkanoate compounds seems to indicate that the oil has undergone some degree of heating, which isn't unusual if the oil was rendered from a marine animal source. The yellow pigment is gamboge. We see some heated pine resin and sterols. In this case, the use of PyGCMS was really important because the mixture also contains some collagen-type protein, as well as gum arabic. These natural polymers need to be broken into smaller pieces to be analyzed by GCMS. The protein may originate from the oil if it wasn't highly purified, or another source. Gum arabic is a common ingredient in watercolor paints, and the present here in an oil paint is intriguing. This example makes the argument for the routine use of PyGCMS for samples. Sometimes FTIR does not indicate that a polymer is present, possibly due to absorbent band interference from other substances or trace quantities. Better to be safe and use PyGCMS to find all the polymers and the non-polymers. So let's talk about data analysis now. Each chromatogram can easily contain 1,000 different peaks. How can you consistently and accurately identify components that are present? For instance, this is the chromatogram from an acrylic gloss varnish. Many of the peaks are found in the literature. Some are in a NIST mass spectral library, but how can we tap into all that information consistently to understand the makeup of the varnish and other complicated samples? A group of scientists from the United States, Netherlands, France, and Canada are developing a tool for everyone to use for PyGCMS investigations. ESCAPE uses free data analysis software that's universally compatible with all GCMS data software, and a mass spectral library that's been donated and curated by experts in the field, and good old Microsoft Excel for the data sorting tool. ESCAPE gives scientists who use it the combined benefit of many experts in the field at identifying and categorizing materials. So ESCAPE takes a complicated mixture like this and identifies the peaks, and then sorts everything into classes. So I used ESCAPE to help identify and categorize the previous acrylic varnish sample, and this is what I got. You see the overall breakdown of the composition, a clear picture of the polymers that are present, and how they're joined in copolymers, such as butyl acrylate, methyl methacrylate, EA, MMA, and even a tiny little tick of methyl acrylate, methyl methacrylate. So ESCAPE provides summary reports that can be saved with the data files or printed off for your clients or conservators. Let's look at it again. How about a more unusual paint? The paint used on an aircraft that was designed in the early Cold War in Canada in the 1950s. This important piece of our history is in the collection of the Canadian Aviation and Space Museum. Using direct inlet PyGCMS at 450, I obtained a chromatogram with many interesting peaks. Then ESCAPE helped me identify important components and determine what kind of paint was used on the aircraft. This paint was chosen because it was hard, smooth, and strong for superior aerodynamics. The ESCAPE tool was able to give me a breakdown of the components so that I could identify it as an epoxy paint based on bisphenol A. Expert knowledge that was written into the ESCAPE template by Nathalie Baucar at CEDA RMF explained which compounds are unique markers for epoxy paints and what is required for a positive identification. Right now, there's 4 versions of ESCAPE that have been developed, and some of you may have heard about or been using some of these already. We have Radical ESCAPE, which was the very first one where it all began for Asian lacquers. Natural ESCAPE for traditional paint media, Synthetic ESCAPE for synthetic paints, which is what I've been using here, and Plastic ESCAPE for plastics. And there's room for more. Where can you learn more about ESCAPE? Join MASC, the users group for mass spectrometry and chromatography. MASC was created in 2003 for the field of heritage science to share information about sampling, sample prep, analysis methods, and data treatment. And I'm excited to announce that MASC will host and support ESCAPE. At the next meeting in 2026, we'll include a day for a dedicated ESCAPE workshop. All of the ESCAPE data analysis files, mass spectral library, and learning materials will be available from that point forward on the MASC website. So please come and join us. And so now I want to thank you all for listening today and for your interest in PyGCMS. It's been a real game changer in my analyses and for what I'm able to identify. If you're already using this tool or someone who's just getting into it, I'm really happy for you. I wish you many enjoyable investigative hours. And please, I want to remind you to reach out if you have any questions. Stay connected. We can achieve so much more as a community. And thank you once again.
Perfect. While we give the audience just a few more minutes to write their questions into the Q&A, as this is the concluding lecture of the 3rd edition of the lecture series Current Topics in Heritage Science, I would like to thank again all the speakers that have contributed to this edition. And thank you also to everyone that joined our lectures. I invite you to visit the E-RIHS website and the social channels to stay updated on future activities.